Welcome back to the home study modules of the dry needling and manual trigger point courses of MyoPain Seminars. This is Jan Dammerholt, President of MyoPain Seminars. In Module 5 we will explore some of the scientific backgrounds of myofascial pain. Although you have already studied several aspects of myofascial pain, we really have not answered the question, what is it? What is myofascial pain all about? In a broad sense, myofascial pain is caused by trigger points, which are hyperirritable spots in the taut band of a skeletal muscle that usually features referred pain. This animation, which is featured on YouTube, shows very nicely how trigger points may develop in skeletal muscles. After overload of the muscle, maybe by eccentric loading or heavy concentric loading, the muscle may start developing contraction knots, which feature shortened sarcomeres within the knot, and because of the tension generated along the muscle fiber, elongated or stretched sarcomeres right outside the knot. When this occurs over multiple closely related muscle fibers, the region may become harder and palpable, which we then clinically can recognize as a myofascial trigger point. While trigger points are very common and myofascial pain is a very common phenomenon, the incidence of myofascial pain syndrome is still difficult to estimate. You may be surprised that muscle is not commonly studied in medical professions, including physical therapy, chiropractic, and others. Particularly when you realize that 42% of the body weight of the average female consists of muscle tissue and 47% of the average male. In a previous home model, we have already quoted Dr. Simons from his article Orphan Organ published in the Journal of Musculoskeletal Pain. According to Dr. Simons, since no specialty claims skeletal muscle as its organ, it is often overlooked. As a reminder, here you see Dr. David Simons receiving his first dry needling treatment from Dr. Joe Donnelly in Atlanta, Georgia. As you may recall, this slide was also featured in a previous home study module. And we talked about the different regions of pain research. Some research is focused on cytokines, others on glia cells, others on peripheral and central nervous system mechanisms. And maybe because muscle has been an orphan organ for so long, the focus on musculoskeletal pain has been delayed and is still fairly young in its development. Of interest here is that pain medicine specialists value myofascial pain much more than other physicians. Both studies in the United States and in Germany show that there is a significant discrepancy in the perceived effectiveness of myofascial treatment between general practitioners and pain specialists. And from the perspective of pain medicine specialists, myofascial pain and trigger points are often the missing link in the overall picture. Several studies have explored at what age myofascial trigger points can first be identified. And it has been shown that infants up to about 1.5 years of age really do not have trigger points. Once children start walking and functioning more in the upright position, trigger points become increasingly common. There is a growing body of literature that supports that with aging, the risk of developing myofascial trigger points actually increases. The late Dr. Leonardo Vecchiet from Italy wrote in 2002 that the increased complaints of musculoskeletal pain with aging appear to be the result of a complex interaction between the physiological process of aging on the one hand, which seem to promote muscle damage and muscle hypersensitivity, and on the other hand, the increased prevalence with age of most potentially painful pathological condition of the muscle itself and other deep somatic structures. According to Dr. Vecchia, this is more common in males than in females. Myofascial pain is definitely one of the conditions that seem to worsen with age. Just a quick reminder that at MyoPain seminars we believe that dry needling and manual trigger point therapy work most likely through biomechanical and neurophysiological mechanisms, just like manual therapy in general. That will also be applied in this current lecture. Unfortunately, the current state of affairs regarding pain science education in physical therapy schools in the United States leaves much to be desired. From this report published in November 2014, we learned that less than 50% of faculty members who responded to a survey were even aware that the Institute of Medicine has issued a report on pain, or that the International Association for the Study of Pain has published guidelines for physical therapy pain education. If you are not familiar with these, the IASP has published guidelines for many different disciplines in healthcare with suggestions what should be included in the education. Only 61% of the respondents believed that their students received adequate education in pain management. That strikes us as very odd. Isn't pain the main reason patients come to physical therapy? So let us take a closer look at some of the underlying scientific evidence of trigger point formation and trigger point issues. The most accepted hypothesis is the integrated trigger point hypothesis, which we will review right now. One of the first things to consider in trying to explain the mechanisms or the formation of myofascial trigger points is that the contracture that houses a trigger point, or the taut band in other words, can in fact block local blood supply to the region of a trigger point. The contracture more or less creates a dam in the river, so to speak, which leads to a retrograde blood flow, and that would mean that the trigger point is actually at risk for not getting enough blood flow and therefore not enough oxygen. In other words, when the taut band or contracture causes a restriction in major arteries, there is a higher outflow resistance. This is a schematic model used in this study from the National Institutes of Health of the United States. In the lower half, the blood flow is normal, and there is almost no restriction in the capillaries. The typical outflow resistance still creates a bit of a vascular bed, but that is mostly because pressure is required to pump blood through a vessel. In the upper half of the picture, the blood flow near an active trigger point is restricted due to the greater resistance from the contractured fibers. As a result of the stagnation, there is a greater blood volume just before the blood flow encounters the area of greater resistance. That would imply that right outside a myofascial trigger point, the oxygen saturation should be much higher than in the region of the trigger point itself. Of course, in reality, blood flow patterns are much more complicated, but for the purpose of this study, this model does suffice. It's rather intriguing that 20 years earlier, so 20 years before the NIH study was published, researchers in Germany already looked at the tissue saturation levels in and near myofascial trigger points. And what they found consistently, that the normal oxygen saturation in muscles was fairly constant and fairly consistent. As the probe came closer to a myofascial trigger point, the oxygen saturation levels actually increased significantly, and upon further moving the probe inside a trigger point, there was a sudden drop to much lower levels. As you can see here, the initial levels are about 30, 40, and in the end it's about 5 maybe. So that's a significant drop, indicating significant hypoxia and maybe even ischemia in the tissues. And just to be sure that we understand the difference, hypoxia is a term to identify deficient oxygen to body tissues. Ischemia is a deficient blood supply. Of course, both end up in a shortage of oxygen where you need it most. Irrespective of the cause of low oxygen supply, anywhere in the body, when the body is depleted of oxygen, the pH of the tissue will drop drastically. With respect to myofascial trigger points, studies again at the National Institute of Health have shown that active myofascial trigger points have significantly lower pH levels than latent trigger points and normal muscle tissue. As you can see in this graph, the pH can drop well below 5, which indicates a very acidic environment, especially at active myofascial trigger points. So, before we further explore the integrated trigger point hypothesis, let's take a little sidestep and take a closer look at what microdialysis really is all about. In a really nice overview article published in 2014 in the Journal of Pain Research, the authors of this paper wrote that microdialysis is an in vivo method for studying local tissue alterations, and that allows for the sampling of substances, and pH in this case, in the interstitium of the muscle, where nociceptor-free nerve endings are found closer to the muscle. We know from several sources, including this article published in Pain, that the extracellular matrix plays a key role in physiological functions of the cell, including the primary efferent nociceptor. So if you can actually measure which chemicals are floating around in the extracellular matrix, it can give you an idea whether the primary efferent nociceptors are going to be activated or not. Microdialysis, and there are many examples of that now, can contribute to the understanding of activated peripheral nociceptive and pain mechanism in humans with chronic pain. Let's take a close look how that may actually be happening. You should know by now that in order to have a pain experience, you need to have an activation of nociceptive nerve endings. In this case, small diameter, slowly conducting efferent nerve fibers from skeletal muscle, in other words, free nerve endings of group 3 or group 4, which are the A delta and C fibers respectively, can transmit this nociceptive information, and they have to be excited before you can experience pain. In the previous home study module, we have given you a definition of a nociceptor. Here we give you a slight variation on the theme, according to the International Association for the Study of Pain. A nociceptor is defined as a high threshold sensory receptor of the peripheral somatosensory nervous system that is capable of transducing and encoding noxious stimuli. During the past 50 years, scientists have learned much about muscle nociceptors. In fact, 50 years ago, no one knew that muscles did have nociceptors, and a lot of the research on pain was actually conducted on the skin. We now know that nociceptors, not just on muscle but in general, can respond to either single or to combination of stimuli, particularly noxious mechanical stimuli, temperature, and chemical substances. In this diagram, you see multiple nociceptors related to muscle, such as purinergic receptors, vanilloid receptors, adenosine receptors, serotonin receptors, and glutamate receptors, amongst others. We have already shared with you that a sensitized nociceptor has a lower threshold for activation and can thus be activated by stimuli that are normally innocuous. It is now known that many chemicals are involved in the process of peripheral sensitization, such as protons, nitrogen oxide, potassium, ATP, nerve growth factor, tumor necrosis factor alpha, interleukin 6, prostaglandin E2, and glutamate, amongst many others. In the illustration, you see a different variation on how some of these receptors are embedded in the cell membrane. It is interesting that several of these substances appear to have both analgesic and sensitization effects. In other words, they can cause local pain, but they can also contribute to peripheral and central sensitization. For example, serotonin, glutamate, and potassium have that quality. Other chemicals may actually have anti-inflammatory roles. Certain cytokines, such as IL-10, IL-4, IL-13, and even IL-1, receptor antagonists, have anti-inflammatory functions. In addition, endogenous opioids, so opioids that the body produces itself, also can have anti-inflammatory functions. The illustration shows how incredibly complex each one of these chemicals is in its function in the body. This is the IL-10 pathway, and if you see how many steps are involved with just this one chemical, you can understand that the real body is so incredibly complicated, and there are actually really very few people who have a grasp of all these details. It gets much more complicated than we share with you in these lectures. And to further illustrate the unbelievable complexity of these mechanisms, there are many other substances that may have anti-inflammatory roles, such as fatty acid metabolites, including prostaglandins, resolvins, protectins, etc. There is, however, a significant lack of good studies. If you look at the metabolism of fatty acids, you can tell it's quite complicated, and that's probably one of the reasons we don't know much about this yet. What is so appealing in microdialysis systems is that it is now possible to measure minute quantities of various chemicals in different tissues. So researchers at the US National Institute of Health, under the leadership of Dr. Jay Shah, developed an ingenious microdialysis system. And you can see it here illustrated. They created a needle with a diameter of only 0.3 millimeter, in which they inserted two tubes connected to a microdialysis pump and a Terasaki plate. In addition to inserting two delivery tubes inside this needle with a diameter of 0.3 millimeters, the researchers also added a semi-permeable membrane at the tip of the needle. If you look at the picture of the needle with the two inserted tubes in relationship to a penny, you can understand that we're now dealing with nanotechnology. The research method has been submitted to the Journal of Applied Physiology to be evaluated in peer review for the validity and reliability. And the Journal of Applied Physiology did publish the paper and concluded that this is indeed a valid method of measuring the chemical environment of different tissues. This animation shows how this needle, which was made specially for the study, can sample the chemical environment in normal tissue close to an active or close to a latent trigger point by sucking up the chemicals found at its very tip. This allows researchers to analyze what the chemical environment is in different parts of the muscle. As we already stated a few minutes ago, this dialysis system also allowed the researchers to measure the pH in different tissues, in this case in normal tissue, near an active trigger point, and near a latent trigger point. And what the researchers established that, especially near active trigger points, the pH is very low, well below 5. So now that we know that active trigger points have a very low pH, let's look at the possible consequences of having this low pH. First of all, anytime the body drops oxygen saturation levels, the pH drops, as we already established. Soon as the low pH is identified by receptors specially made for measuring low pH, the nervous system starts releasing several nociceptive substances, such as you see listed here, CGRP, or calcitonin gene-related peptide, bradykinin, ATP, serotonin, and prostaglandins. Unfortunately, any of these substances can lead to more nociceptive input and therefore eventually possibly to the experience of pain. The low pH actually does more than just that. At the motor end plate, low pH will downregulate acetylcholine esterase, which means that once acetylcholine is released from the nerve terminal into the synaptic cleft, the acetylcholine esterase is not there to break down this acetylcholine, resulting in excessive amount of acetylcholine available in the cleft. The body has several nociceptors sensitive to low pH, such as acid sensing ion channels and transient receptive phenyloid receptors, and some others as we see in just a minute. Once the low pH activates these receptors, this will lead to hyperalgesia, which is a sign of central sensitization, and the release of inflammatory mediators. So let there be no misunderstanding. As Kathleen Sluker, a researcher physical therapist in Iowa, has demonstrated many times again and again, an acidic pH has a profound effect on the initiation and perpetuating of muscle pain. The more acidic a milieu may be, the more it will activate these acid sensing ion channels, particularly 1 and 3. These are muscle nociceptors, which in turn can produce mechanical hyperalgesia. If we apply this to the trigger point story, we know that in the vicinity of an active trigger point, the contracture has blocked the local blood flow, which leads to a lack of oxygen, which leads to a release of all these neurochemicals, a low pH. The low pH can produce mechanical hyperalgesia. In other words, a trigger point region can become very sensitive. This does not mean that we're dealing with circular reasoning, as some opponents to the trigger point concept have suggested. That is a clear misunderstanding of what actually happens. As we have seen already, the pH in active trigger points is well below 5. In studies of rabbits, when the intracellular pH dropped from 7 to only 6.6 during 4 hours of ischemia, the drop in the pH was more than enough to fully activate the nociceptive ASIC3 channels in nearby neurons. Therefore, we can be assured that with a pH below 5, these nociceptors are activated and contribute to the mechanical hyperalgesia. It probably does not come as a surprise that the story once again is much more complicated. We have multiple other possible receptors for low pH, as you see here. We have more vanilloid receptors, but we also have short transient receptor potential channels. This goes way beyond what you need to know for the courses in trigger point dry needling. It's just provided as an illustration. The microdialysis study from Dr. Shah and colleagues at the National Institutes of Health revealed that in active trigger points the concentration of calcitonin gene-related peptide is much higher than in latent trigger points on normal muscle tissue. When they performed a dry needling procedure, the concentration of the calcitonin gene-related peptide immediately dropped close to normal levels, which is significant and may indicate that dry needling can actually modulate the chemical environment of active trigger points. Similar observations were seen for substance P, as you see in this slide. Elevated levels were observed in the neighborhood of active myofascial trigger points for norepinephrine, tumor necrosing factor alpha, interleukin 1, 6, and 8, and serotonin. Based on personal communication, interleukin 12 levels were reduced. While these studies are very important and change the hypothetical thinking about myofascial pain and trigger points, we should note that these studies had indeed very small sample sizes. There's also a potential technical issue with the studies because to create a local twitch response, they actually used a dialysis needle. Which brings up the question, how did the researchers make sure that the needle ended up at the exact same place as before they moved the needle to create a twitch response? I frankly do not know the answer. I have been personally told that they controlled for that, but the studies do not necessarily reflect how that was accomplished. In 2012, another important study was published that also evaluated the presence of biochemicals in the close vicinity of trigger points. This study was performed on rabbits, and they used a very different technology. They measured some of the same substances, such as substance P and tumor necrosing factor, but also several others, as you see listed in this slide. All these chemicals are involved with pain, inflammation, and hypoxia. What the researchers confirmed is that dry needling can indeed modulate the concentration of these chemicals. But as they found out, it was dosage dependent, and that is an important variable. With relatively easygoing dry needling, the chemical concentrations decreased significantly. However, when the intensity of the dry needling was increased significantly, the chemical concentrations actually went up. And that is a very important observation. Of course, this study was performed on rabbits, and we always have to be careful transferring this to human subjects. And that study really should be done at some point. But it is conceivable, based on this study and the previous one, that when dry needling is performed at some other random method, when the needle is used as a search tool to find the trigger point, it is very easy to understand that the dry needling could be too intensive and actually increase the concentration of the chemicals and therefore worsen the patient's subjective pain. Therefore, we advocate, as we do in all our courses, to really palpate very carefully, find the trigger point manually, and then do the least amount of dry needling possible, but enough to change the chemical environment, and by doing so removing potentially nociceptive input that can aggravate the nociceptors. I think it is fair to conclude that the studies actually show significant differences in the pH and in the concentration of the chemical substances in the immediate vicinity of active trigger points. And this seems to change with the local twitch response elicited by a needle. I should also note that acetylcholine and glutamate have not been measured in direct relationship to trigger points. There are, however, several other studies that have measured glutamate levels in patients with myalgia. So while these studies did not specifically look for trigger points, in their study of myofascial temporomandibular disorders, for example, significantly higher glutamate levels were found in a patient group compared to a control. And here you see two other studies that reported an increased interstitial muscle concentration of glutamate in trapezius muscles with myalgia. They also found that the interstitial muscle concentration of serotonin in the trapezius was significantly increased. So when we try to summarize some of the key aspects of the integrated trigger point hypothesis the way it stands now, we assume there is some level of muscle overload, and that could be from acute or repetitive activities, sustained contractions, or even low threshold fiber activities. We'll go back to the Cinderella hypothesis. All these can lead to ischemia and hypoxia, which on the one hand reduces the pH, therefore increasing acidity, but it also leads to an inhibition of acetylcholine esterase on the left, and it can lead actually directly to muscle injury. If we go back to the mid-track, when the pH is reduced, it will trigger the release of lots of chemicals, and in this diagram we only mentioned hydrogen ions and CGRP, or calcitonin gene-related peptide, but as you know by now there are many others. CGRP was selected because it has such a profound influence on many aspects of the chain. CGRP on the one hand lowers the concentration of acetylcholine esterase, but it also increases the release of acetylcholine from the nerve terminal. And it also can lead to an upregulation of the nicotinic acetylcholine receptors on the other end. The net result is that there are increased motor end plate potentials. That leads to an increase of actin and myosin interactions, therefore contractures. That leads to sarcomere contractions that eventually becomes the taut band on the one hand, but it also leads immediately to pain. And that can be local pain, referred pain, and eventually peripheral and central sensitization, as we will see in a future lecture. Back to the left of the screen, the taut band can also add to the sympathetic drive. There may be more axonal activity and therefore an increase of acetylcholine at the nerve terminal. We've looked already at some of the adrenergic receptors that may play a role at the motor end plate, although that's not totally known. The taut band in itself is also a form of muscle overload, and therefore the system is capable of maintaining itself. Multiple studies have confirmed that the dysfunction of the motor end plate can be visualized as end plate noise. What you see here, if the electrical activity is measured directly in a trigger point, you see this consistent pattern of electrical discharge. Initially this was called spontaneous electrical discharge, and later it was concluded that this really is end plate noise. If you go a centimeter or so away from the trigger point in the same muscle, you really do not see the same level of activity. So myofascial trigger points can now objectively be identified with a monopolar Teflon-coated EMG needle, identifying this end plate noise. Another important finding is that the prevalence of the end plate noise in trigger point regions was significantly higher in active trigger points than in latent trigger points, and the pain intensity and the pressure pain thresholds were also highly correlated with the prevalence of end plate noise in the trigger point region. So the more end plate noise, the more patients complained about pain. So end plate noise may actually be an indicator of the pain intensity. Another feature of trigger points is that the muscle never really relaxes. What you see in this slide is a muscle activation and an EMG reading on the right of non-trigger points. either with indwelling EMG or with surface EMG, you see there is a distinct beginning and an end of the electromyographic reading. On the left of the slide, in the trigger point region, there is a constant discharge in both the intramuscular EMG and the surface EMG, and that indeed is that spontaneous electrical activity or end plate noise. While end plate noise is not unique to trigger points, it certainly has become a characteristic of active and latent trigger points. And there really is no question that this end plate noise can be objectively measured without any placebo or any other aspect of that. As you can see here, it has been identified in horses, and horses really don't do much with placebo. There are also several rabbit studies that show this end plate noise. The study that's quoted here actually showed that the end plate noise does not change when you either cut the spinal cord or the peripheral nerve. That indicates that the end plate noise and the dysfunction is really something that happens at the motor end plate itself and is therefore a fairly local phenomenon. We could provide you with many more references, but here are two additional references from 2001 and 2002 that also discuss and show that these end plate potentials are very common to myofascial trigger points. If trigger points are indeed the result of an excessive release of acetylcholine from the motor end plate, it follows that injections with botulinum toxin should be helpful in the management of patients with myofascial pain. In our clinic, when we refer patients to a local physician who does botulinum toxin injections at a very high level, we find very, very good results. Yet nearly 50% of the published studies do not support the use of botulinum toxin for myofascial pain. In the diagram here you see what the action is of botulinum neurotoxins, but in the next slide you see a video that explains this probably a little bit easier.
Botulinum toxin type A binds to the motor nerve terminal. The binding domain of the type A molecule appears to be the heavy chain, which is selective for cholinergic nerve terminal. The toxin binds with high affinity to the motor neuron. Botulinum toxin type A is internalized via receptor-mediated endocytosis, a process in which the plasma membrane of the nerve cell invaginates around the toxin receptor complex, forming a toxin-containing vesicle inside the nerve terminal. After internalization, the light chain of the toxin molecule, which has been demonstrated to contain the transmission-blocking domain, is released into the cytoplasm of the nerve terminal. Botulinum toxin type A blocks acetylcholine release by cleaving SNAP25, a cytoplasmic protein that is located on the cell membrane and that is required for the release of this transmitter. The affected terminals are inhibited from stimulating muscle contraction. The toxin does not affect the synthesis or storage of acetylcholine or the conduction of electrical signals along the nerve fiber. Evidence indicates that chemical denervation of the neuromuscular junction by botulinum toxin type A results in an expansion of the end plate region and growth stimulation of collateral axonal sprouts. A nerve sprout eventually establishes a new neuromuscular junction, and muscle activity gradually returns. Recent research suggests that this new nerve sprout eventually retracts as the original junction returns to functionality. The sprouts disappear and the original end plate returns to full activity. In either case, repeat injections of Botox-purified neurotoxin complex may be required to maintain the desired clinical effect.
To explore why there is such a discrepancy in botulinum toxin studies in the treatment of myofascial pain, Dr. Robert Gurin wrote an excellent analysis of the literature on this topic. And indeed, few studies have been designed to avoid many of the pitfalls associated with the trial of botulinum toxin treatment trigger points. Dr. Gurin identified several common problems that are very closely related to each other. In some cases, the studies picked the wrong or confounding or inappropriate control populations or treatments. If the treatments are not that different from botulinum toxin, or if the control populations are very closely to the botulinum toxin populations, you will not see significant changes between the experimental and control groups. It could be that the placebo responders are so robust that they kind of cloud the outcome of the botulinum toxin. In some of the studies, the treatments were incomplete. In other studies, there was a misinterpretation of the time frame of action of botulinum toxin. It is often thought that botulinum toxin may take about 2 weeks before its full effect is noticeable. So if you measure the outcomes in 4 days or 5 days, you are not going to see the optimum outcome from the botulinum toxin. This is a brilliant paper, and we provided you with a copy of it. The integrated trigger point hypothesis, originally formulated by Simons and colleagues, included an autonomic function without going into much detail. In our 2004 paper, an expansion of Simon's integrated hypothesis of trigger point formation, we mentioned that at the motor end plate there are alpha and beta adrenergic receptors. And these receptors could possibly provide a mechanism for the autonomic interactions. We know from rodent studies that when these adrenergic receptors are stimulated, it actually adds to the release of acetylcholine in the phrenic nerve. We are not aware of any studies in humans, but it is conceivable that even in humans, when the, for example, alpha adrenergic receptor gets activated, that this could be a extra factor for the spontaneous release of acetylcholine. In summary, the integrated trigger point hypothesis proposes that there is an abnormal depolarization of the post-junctional membrane of motor end plates, which causes a localized hypoxic energy crisis, which is in turn associated with sensory and autonomic reflex arcs that are sustained by complex sensitization mechanisms. The presence of spontaneous electrical activity or end plate noise, and the clinical evidence that treating trigger points eliminates or significantly reduces this end plate noise supports the notion that trigger points are located in close proximity to dysfunctional motor end plates. An elegant study by Barbero and colleagues showed that the innervation zones and trigger points are indeed located in well-defined areas in the upper trapezius muscle. At this point, we're going to spend a few minutes looking at a study by Cheng, Chen, and Chang about neuromuscular jitter and trigger points. Let me first try to explain what neuromuscular jitter is all about. If we imagine an alpha motor nerve on its way to a muscle, right before it comes close to the muscle, it will branch off in multiple branches, each with its own motor end plate. In an optimal muscle contraction, the electrical signal should reach these motor end plates at the same time, causing the release of acetylcholine at the exact same time, and therefore have a depolarization at the exact same time, and therefore have an optimal muscle contraction. So in other words, as it says here, the neuromuscular jitter is produced by fluctuations in the time for end plate potentials to reach the threshold for action potentials. So in other words, each one of these branches may not quite get the electrical signal at the exact same time. These fluctuations in time are called neuromuscular jitter, or they cause neuromuscular jitter. With a dysfunctional neuromuscular junction, muscle fibers of the same motor unit may not always fire in the same sequence that causes jitter. Here you see a graphical representation of how these electrical signals at the various branches of a motor nerve can be out of sync, creating jitter. This is from the excellent textbook on electromyography by Jo Fransen from the Netherlands. And what they found in this study that patients with trigger points had significantly more jitter, or as it's also referred to, increased mean consecutive difference in the trapezius and levator scapulae muscles compared to controls. So the authors concluded that at least part of the end plate dysfunction may be the result of a disintegration of spinal motor neurons. And they said the degeneration of these motor neurons may trigger sensory nerve involvement and hypersensitivity to pain. The researchers found a positive correlation between jitter and the duration of myofascial pain, and from their perspective they claim that it supports the development of progressive neuronal degradation with axonal neuropathy in more chronic cases of trigger points. That's an interesting perspective, and it assumes that this is a feed-forward problem. However, as I mentioned already, when we cut the peripheral nerve in a rabbit, the problem does not go away. So it's hard to imagine that it is always the feed-forward system, and it could really be just a local problem at the end of the muscle motor end plate. Here you see the electrical activity before transaction of the spinal cord and 30 minutes after transaction of the spinal cord, and there's very little difference. Even 60 minutes after transaction, there is no difference. The same occurred when the peripheral nerve was cut, and 30 minutes later, the electrical activity had not changed. So, as it does not change the electrical activity, it argues kind of against this progressive neuronal degradation idea with axonal neuropathy. It is conceivable when the muscle junction and the muscle are already dysfunctional, in other words, when there is a contracture or a taut band in the muscle, maybe the muscle cannot receive the information to lead to cell depolarization. We don't know that for sure, and that's somewhat speculative, but it does argue against this progressive neuronal degradation idea. In a more recent study from 2011, the same authors and one additional author confirmed again that the jitter values, or mean consecutive difference values, obtained from trigger points and trapezius were significantly greater than controls. This really indicates that there is a motor control problem in the presence of trigger points. In fact, 71.2% of the patients had abnormal jitter in the trapezius muscles. And again, they confirmed a positive correlation between the MCD or jitter values and the time that people had myofascial pain. So the longer people had myofascial pain, the more problems were observed in the motor control as expressed by jitter. And that concludes Module 5.